I told you at the beginning of the chapter that the reproduction occurs through few events, right? The first event in the reproduction is gametogenesis, means formation of sperm and ova, that we already completed in spermatogenesis and oogenesis, right? Even we discussed in the last lecture, which one? Menstrual cycle, by which how a female prepare her uterus to give birth to a baby, to provide space for the growth of the baby, right? The next step is followed by gametogenesis is insemination. After that, fertilization, formation of the zygote, it will continue. So already gametes are formed. The next step we have to discuss is fertilization. But before going to fertilization, as we know very well, it all begin with which one? Insemination. Once gametes are ready, the next step is insemination, is the process of deposition of a semen into the vagina of the female by the male partner is called insemination. Once semen or the sperms are deposited in the vagina, the sperms, millions of sperms are there. We know that per ejaculation will contain 2.5 to 5 ml of semen, which contain about 200 to 300 million sperm. Many sperms will start their journey, and while reaching ampullary region, approximately, while starting millions are there, while reaching ampullary region, hardly 100 sperms are able to reach it. Among 100, only one sperm will be finally successful in carrying out which one? Fertilization. In human, the ova is fertilized by only one sperm, and the condition is called monospermy. What is monospermy? Fusion of a ova by single sperm. Okay. What is that? But there should be no polyspermy. What is polyspermy? Fertilization of a ova by more than one sperm, right? This condition should not happen in our case. There will be only one sperm should be able to fertilize it. Now you may ask me why there is no polyspermy, why it should not occur. Very simple. The ova has, right, n haploid number of chromosome, that is 23 chromosomes are there. A sperm is also haploid, that also has 23. 23 plus 23 during fertilization, it becomes 46. Suppose if one more extra sperm fertilize, 46 plus 23. Can you imagine? Remember that in humans, if the chromosome number from 46 increases just by one chromosome, not 23, 46ರಿಂದ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಜಾಸ್ತಿ ಆಗಿ 47 ಕ್ರೋಮೋಸೋಮ್ ಆದರೆ ವಿ ಹ್ಯೂಮನ್ಸ್ ಸಫರ್ ಫ್ರಮ್ ಡಿಸಾರ್ಡರ್ ಲೈಕ್ ಡೌನ್ ಸಿಂಡ್ರೋಮ್, ಕ್ಲೈನಿಫೆಲ್ಟರ್ ಸಿಂಡ್ರೋಮ್ ಅಂತ ಬೇರೆ ಬೇರೆ ಸಮಸ್ಯೆಗಳು ಬರಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗ್ತವೆ. ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಜಾಸ್ತಿ ಆದ್ರೇ ಹ್ಯೂಮನ್ಸಲ್ಲಿ ಎಬ್ನಾರ್ಮ್ಯಾಲಿಟಿ ಫಾರ್ಮೇಷನ್ ಆಗುವಾಗ, ಕ್ಯಾನ್ ಯು ಇಮ್ಯಾಜಿನ್ ಇಫ್ 46 ಕ್ರೋಮೋಸೋಮಿಗೆ ಝೈಗೋಟಿಗೆ ಇನ್ನೊಂದು ಎಕ್ಸ್ಟ್ರಾ ಸ್ಪರ್ಮ್ ಆ್ಯಡ್ ಆದರೆ ಪ್ಲಸ್ 23, can that zygote tolerate? No. The embryo or the zygote will die. It cannot tolerate. That's why after it, only what condition should be occur? What condition should be seen? Monospermy. There should be no polyspermy. Even though 100 sperms will reach, only one sperm will be able to fertilize this. Now, the next very important thing. Now, the semen is deposited. Sperm started their journey. Among them, imagine the one sperm is a very fast runner like a Usain Bolt. Okay, one or two sperm. Within few minutes of journey, okay, or their deposition, they reached ampullary region and they happily said, yes, I have reached. The question is, are they able to fertilize? No. Very, very important thing, after insemination, just before fertilization, they have to undergo a process called, the sperm has to undergo a process called what? Capacitation. What is capacitation? Name itself tells, by this process sperm will get a capacity to fertilize. Right? As the sperm start their journey, right, while reaching ampulla and just before starting the fertilization, the sperm has to undergo capacitation. The entire capacitation process takes around 7 hours. Okay, 7-hour capacitation process, during which some cholesterol content present on the head of the sperm. Try to understand, sperm head is covered by some cholesterol content and other things are there. Tumba detailed study avashyakate illa. Those cholesterol content from the head of the sperm are removed during capacitation. Sperm get the capacity to fertilize. For that, they have to spend how many hours? 7 hours. So, immediately after sperm deposition, within few minutes, if sperm reaches the ova or the ampullary region, bandu reach adru kuda, it is unable to fertilize. It has to spend how many hours? 7 hours. Okay. Now, one more question that I asked you long back, right? Millions of sperms are deposited. Only 100 sperm will reach. Only one sperm will fertilize. Question is, what is the need of deposition or the production of millions of sperm? Ishtu sperm yake deposit agbeku alwa? The answer is, just for your reference, it will not ask in the exam. Remember, from the site of insemination to the site of fertilization is about 30 centimeter. From insemination to the site of fertilization, 30 centimeter. Sperm will travel with a speed of 3 millimeter per minute. Okay. Now, among the millions of sperms, okay, which started their journey, this journey up to ampulla is a very tiring one. It makes the sperm very tired, because of which many sperms among the million will die. It is a tiring journey. Many sperms will die in between. They are unable to reach. Okay. Number 2, the second reason is, this vagina is acidic in nature. I already ಹೇಳಿದ್ದೇನೆ ಇದರ ಬಗ್ಗೆ. So what happens, even though bulbourethral gland fluid is released first, which neutralizes acidic nature, but still due to the acidic effect, again many or large number of sperm will die. The acidic nature of the vagina kills several sperm. Sperm has started its journey, okay, just in a belief that I have fructose with me. Fructose present in the seminal vesical fluid, which is a source of energy. It thinks that only fructose is there, I can reach the target site. But in several thousands of sperm, fructose gets exhausted. Fructose khali agi bitre, avaru in between the race enagbodu, die ಆಗ್ತಾರೆ. Several sperms will, thousands of sperm will die. Sperm count decreases. Now, during their journey, as uterus is an organ which has macrophages. Hope you know what are macrophages, alva? Bodyಯ ಎಲ್ಲ ಟಿಶ್ಯೂಗಳಲ್ಲಿ macrophage ಇದೆ. ಇಲ್ಲಿ ಕೂಡ ನೀವು macrophage ಅನ್ನು ನೋಡ್ತಾ ಇದ್ದೀರಿ. These macrophage will eat few sperms, because of which also sperm count decreases. Another reason, among them many sperm will reach wrong fallopian tube. Instead of going to the left fallopian tube, they don't know whether the ova is blowing the whistle. Illa, ಯಾವುದೇ interaction illa. Sperm has blindly started its journey with a force of fructose energy. Okay, some sperms will reach wrong fallopian tube. As there is no ova, they will die there only. Because of all these reasons, while reaching ampulla, hardly 100 sperms are able to reach. Suppose deposit ಮಾಡುವಾಗಲೇ 100 sperm deposit ಆದರೆ ಅವರೆಲ್ಲರೂ ಈ ಸರ್ವಿಕ್ಸ್ ಅನ್ನೇ ಕ್ರಾಸ್ ಮಾಡಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. Hope the answer is clear to you. Yes, now let me proceed. So After insemination, sperm will undergo capacitation. Among 100 sperm, only one sperm is able to carry out which one? Fertilization. Then what is fertilization? It is the fusion of haploid male and female gamete, haploid male and female gamete to form a diploid zygote is called fertilization. Next, the fertilization process is possible only when the sperm and ova are transported to ampullary region simultaneously. Mark this point, very important. I repeat, fertilization is possible only when sperm and ova reach ampullary region simultaneously. ಎಕ್ಸಾಕ್ಟ್ ಅಲ್ಲದಿದ್ರೂ ಕೂಡ ಇವರಿಬ್ಬರು ಆಂಪುಲರಿ ರೀಜನಿಗೆ ಆಲ್ಮೋಸ್ಟ್ ಸೇಮ್ ಟೈಮಲ್ಲಿ ರೀಚ್ ಆದರೆ ಮಾತ್ರ ವಿಚ್ ಪ್ರೋಸೆಸ್ ಇಸ್ ಪಾಸಿಬಲ್? ಫರ್ಟಿಲೈಸೇಷನ್ ಇಸ್ ಪಾಸಿಬಲ್. ದಿಸ್ ಈಸ್ ದ NCERT ಸ್ಟೇಟ್ಮೆಂಟ್. ಲೆಟ್ ಅಸ್ ಕ್ರ್ಯಾಕ್ ವೈ ಇಟ್ ಈಸ್ ಸೋ. ಯಾಕೆ? ಇದು ನಿಮಗೆ ಗೊತ್ತಿರಬೇಕಾದ ವಿಚಾರ. ಯಾಕೆ ಅಂತ ಕೇಳಿದರೆ, ಆಫ್ಟರ್ ದ ರಿಲೀಸ್ ಫ್ರಮ್ ದ ಓವರಿ, the ova or the secondary oocyte can survive for just 24 hours. After insemination, deposit ಆದ ಮೇಲೆ sperm can survive for 48 hours. That's why in between, in that period, if both reach, what could be possible? Fertilization can be possible. Ova has reached. If sperm reached late by 24 hours, what happens? There is no possibility of fertilization. Clear? Now, what happens during the time of fertilization? Imagine this is the ova. I have told you the diagram or the structure of it. It is its nucleus. It is covered by zona pellucida. There is a perivitallin space. Around it, there is a layer of corona radiata, nothing but granulosa cells. Okay, layer of granulosa cells are present around it, right? This is zona pellucida, glycoprotein layer. Okay, now as sperm reaches here, I will show you step by step. As sperm reaches here, point number 1, acrosomal enzymes are released. I have told you few different types of acrosomal enzymes like hyaluronidase, corona penetrating enzyme, zona lysins are released. These enzymes gradually dissolve corona radiata and zona pellucida. The enzymes of the acrosome dissolve the corona radiata and zona pellucida, due to which the sperm comes in contact with what? Plasma membrane of the ova. Once a sperm come in contact with plasma membrane of the ova, few changes occur that I will tell you later. Okay, now what happens? The entire sperm will be swallowed inside, or the entire sperm will enter inside. Some authors say only nucleus is released. It is wrong. Remember, in human, the entire sperm will enter inside. In some rare cases, a small part of the tail can be left out. Now, this entire process, once sperm enter inside, gradually the remaining part of the sperm, including the middle piece, tail, plasma membrane, everything will be dissolved, and later the nucleus of the sperm mixes with nucleus of the ova and becomes a diploid cell. This diploid cell, we call it as what? Zygote. And the very important notable point is the zygote is still covered by, zygote ಈಗಲೂ ಕೂಡ ಯಾವುದರಿಂದ ಕವರ್ ಆಗಿದೆ? zona pellucida. I repeat the steps involved in fertilization. Very simple. First, acrosomal enzymes are released. which dissolve corona, radiata and zona pellucida. Plasma membrane of the sperm come in contact with plasma membrane of the ova. Next line, the entire sperm will enter inside the cytoplasm of the ova. Right? Now, as a sperm enter inside, the oolemma or the plasma membrane of the ova will depolarize. I repeat, as sperm enter inside, the plasma membrane of the ova depolarize. Why? To prevent polyspermy. The depolarized oolemma prevent the entry of additional sperm. 99 other sperms are also trying to enter inside. The depolarization and the changes that occur in the zona pellucida prevent what? Polyspermy. Next line, everything of the sperm will be dissolved except haploid nucleus and proximal centriole. Next line, nucleus of the sperm mixes with nucleus of the ova and it forms a diploid nucleus. Now this ova becomes what? Zygote. This single cell is called zygote. Zygote is still covered by zona pellucida and corona radiata. It is all about fertilization. Now this zygote gradually starts its growth, attached to the wall of the mother's uterus and completes its 9 month. After 9 months, the baby will be delivered out. Now the question is that baby will be male or female. That means my question is how sex of the baby will be determined. 5-mark question. Sex determination in human, right? How the baby's sex will be decided. Try to understand. As we know, we have how many chromosomes? 46 chromosomes are there in every cell. The 46 chromosomes can be classified into 2 types. What are they? Out of that 44 autosomes, and the 2 are called allosomes, or they are also called sex chromosomes. And the sex chromosomes are, one is X, another one is Y. In human, a male has 44 autosome plus XY chromosome, eradu kuda ide, whereas in a female it is a basic thing that you know well, 44 autosome plus 2 X chromosomes are present in the female. I will write this condition once again. If you look at the father and mother, the chromosomal condition is, in case of father is 44 autosome alva, 44 autosome plus ತಂದೆಯಲ್ಲಿ ಏನಿದೆ 2 sex chromosome ಯಾವುದು XY, whereas in the mother 44 autosome plus 2 X chromosomes are present. During reproduction, first event is the formation of gametes, nothing but gametogenesis. The father, the sperm which are produced, sperm are of 2 types, in which 50% of the sperms are always haploid. The parental spermatogonia is diploid, spermatozoa is haploid. 4 sperm formation ಆಗ್ತಾ ಇದೆ. 4 ಅಂತ ಹೇಳಿದ್ರೆ 50% ತೆಗೆದುಕೊಳ್ಳುವ 2 plus 2 means 1 plus 1 ಅಂತ ತೆಗೆದುಕೊಂಡ್ರೆ 50% sperms contain 22 autosome plus X chromosome. Remaining 50% sperms contain 22 autosome plus Y chromosome. That means 50% sperm has X, 50% sperm has Y chromosome. Whereas at the end of the oogenesis, the one ova which is formed, it has 22 autosome plus X chromosome. What happened to remaining 22 autosome and X? That has moved in a tiny cell called polar ಬಾಡಿಯಲ್ಲಿ ಹೋಗಿ ಡಿಜನರೇಟ್ ಆಗಿದೆ ಅಂತ ಹೇಳಿ ಅಸ್ಯೂಮ್ ಮಾಡುವ. ನಾವು ವಾಟ್ ನೆಕ್ಸ್ಟ್? ಸ್ಪರ್ಮ್ ಮತ್ತು ಎಗ್ ರೆಡಿ ಇದೆ. ನೆಕ್ಸ್ಟ್ ಈಸ್ ದ ಫರ್ಟಿಲೈಸೇಷನ್ ಆ್ಯಂಡ್ ಫಾರ್ಮೇಷನ್ ಆಫ್ ದ ಬೇಬಿ. ಸಪೋಸ್ the egg having X chromosome is fertilized by sperm having X chromosome. What happens in the child? It becomes E nucleus ಮತ್ತು ಓವಾದ ನ್ಯೂಕ್ಲಿಯಸ್ ಫ್ಯೂಸ್ ಆದರೆ 44 ಅಟೋಸೋಮ್ ಪ್ಲಸ್ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡಿ XX ಅಲ್ವಾ, the baby will develop into male or female. The baby will grow into a female. Suppose e sperm ಬರುವ ಬದಲು ಅಲ್ವಾ, by chance if this ova e sperm ಬರುವ ಬದಲು e sperm ಬಂದ್ರೆ ಅಲ್ವಾ, sperm having Y chromosome, what happens? The baby is going to have 44 autosome plus XY chromosome. That means the baby will develop into what? Baby will grow into male. What I am trying to tell you here is the sex of the baby will be determined when? Determined at the time of fertilization itself. ಜ್ಞಾಪಕ ಇಟ್ಕೊಂಡಿರಿ, fertilization ಆಗಿ ಯಾವಾಗ zygote formation ಆಗಿದೆ, that moment of time only, after 9 months, the baby that is going to come out of the mother womb, it should be male or female. It is decided at the time of fertilization itself. Once fertilization is over, zygote is formed, sex of the baby will be decided. After that, no food, no medicine can change the sex of the baby. ಕೆಲವರು ಹೇಳ್ತಾರೆ ಮ್ಯಾಂಗೋ ತಿಂದರೆ ಹುಟ್ಟುವ ಮಗು ಗಂಡು ಮಗು ಹುಟ್ಟುತ್ತಂತೆ ಅಂತ ಹೇಳಿ. Whether it is true, not mango, not any fruits can change the sex of the baby. It is already decided. Is that clear? And in this case, which partner play a main role in deciding sex of the baby? It is the father or mother? Of course, father. But mother is blamed for giving birth to a female baby, alwa? Whether it is right? No, it is a misconception. Here, ತಾಯಿ ಏನು ಪ್ರತಿ ತಿಂಗಳು ಓವಾ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದಾಳೆ, that ಓವಾ ಆಲ್ವೇಸ್ ಕಂಟೈನ್ ವಿಚ್ ಕ್ರೋಮೋಸೋಮ್ X. ಅವರಲ್ಲಿ ಬೇರೆ ಚಾಯ್ಸೇ ಇಲ್ಲ, ಓನ್ಲಿ X. ಆದರೆ ಆ ತಂದೆ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವ, ಮೇಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವ ಸ್ಪರ್ಮ್ ಏನು ಇದೆ ಅದರ ಒಳಗಡೆ 50% ಆಫ್ ಸ್ಪರ್ಮ್ ಹ್ಯಾಸ್ X, 50% of sperm has Y chromosome. It is totally depending on sperm of the father, not on the mother's ova. So, yava sperm, which sperm fertilize the ova? Based on that, the sex of the baby will be decided. If a sperm having X chromosome fertilize the ova, baby will grow into female. If a sperm having Y chromosome fertilize the ova, baby will grow into male. So, the establishment of sex at the time of fertilization, we call it as sex determination. Male parent plays a dominant role in deciding sex of the progeny.